Can you hear me and can you see the slides properly?
Yes, everything is fine.
Thank you very much for the kind introduction, Ivo. Thanks to the organizers. As Ivo mentioned, I'm kind of, Alan Orwell had obligations today and he couldn't make it, so he asked me to take over, and I would like to present the current work we are working on with him in a collaboration. And the title of my talk is X-ray-free electron laser studies reveal O2 binding to isopenicillin enzyme synthase induces correlated motions during catalysis. 2OG oxygenases, or the 2OG oxygenase superfamily, typically catalyzes hydroxylation reactions coupled to 2OG turnover. So we have our 2-oxyglutarate here, which is turned over to succinate, and we release CO2, and your substrate is hydroxylated. And these enzymes of the 2OG oxygenate superfamily are very widespread in also in humans, and play important roles in collagen biosynthesis, lipid metabolism, nucleic acid repair, and signaling. And as a few of you are probably aware, the work on cells and their how they deal with O2, and how the enzymes, which is in this case PHC2, how they deal with the hypoxic response, were awarded with the Nobel Prize of Medicine and Physiology in 2019. And we are really interested in how oxygen binds to these enzymes, and how what effects we observe throughout the crystals when oxygen enters the active site. I'm focusing now on a member of this 2OG oxygenase fold family, which is called isopenicillin N synthase. And isopenicillin N synthase is catalyzing this reaction, and uniquely, it's not using 2-oxyglutarate, but just oxygen, and using the full potential of oxygen to oxidize to this really constrained ring system. So first forming the beta-lactam ring here, and then the thiolidine ring afterwards. And all clinically used penicillins and cephalosporin antibiotics are later derived from these precursors, so IPNS is still of fundamental importance. And we are really interested in the reaction mechanism, and we had great talks from Diana and Arvin and from people before who were mentioning already that we are trying to make a video basically of our reaction mechanism. And first of all, we have our iron 3 superoxo species. This is the first key intermediate in the reaction cycle. And then there is an iron 4 intermediate also in the reaction cycle on the way to the really constrained IPN product. And we ask ourselves now, how does oxygen bind to IPNS? And how does IPNS control the conformations of intermediates to catalyze the formation of these really strained beta-lactam and thiolidine ring systems? And our approach is using the described XFEL time-resolved 0 femtosecond crystallography methods to monitor the reaction of the anaerobic IPNS ACV complex with O2 to form the IPN complex. What do we need for this? Obviously, we need first crystals, we need microcrystals, and here are two crystal morphologies shown of IPNS. The first one is the hexagonal morphology, and the other one is the needle morphology. We are able to crystallize both and also change the size of the crystals by changing our conditions. But for our studies I present here, we just use the needle morphology crystal form. And yeah, and we have heard about, in Diana's talk, about several methods, and I'm actually focusing today on the AD tape drive method. It's a drop-on-demand sample delivery system, and in this method we have a tape drive and an acoustic droplet ejection system. The acoustic droplet ejection system emits nanoliter droplets with where our crystals are in, and places them on a belt drive. And this belt drive is now moving the crystals throughout the reaction chamber. In the case of our setup, we fill this reaction chamber with oxygen, and then oxygen diffuses into the nanoliter droplets. And by changing the belt speed drive, we can then have a look and change the reaction time. So faster belt speeds are shorter reaction times than the other way around. As soon as the crystals are reaching the X-ray action zone, we are collecting, on the one hand, time-resolved zero diffraction data, and on the other hand, also the emission spectroscopy. And here is a photo showing nicely the IPNS crystals lined up from an LCLS beam time in 2018. And just to have an idea about which dimensions we are talking about a dimension of 2 millimeter. So this diameter is just 2 millimeter. This kind of shows how small these droplets are. And as mentioned before, the complementary method, the simultaneous collection we are doing from each drop, is the X-ray emission spectroscopy. In the X-ray emission spectroscopy, a core electron is excited by an incident X-ray photon, and this core hole, which is created in the 1s shell, is then filled by an electron from a higher state. So in the case of the iron K alpha fluorescence lens lines, we kind of have electron falling back from 2p into the A1s, and in the case of the K beta, we have a electron falling back from the T3p state, the 1s, and these these X-ray emission spectroscopy is really sensitive and really atom specific, and we can also look at the oxidation states. And we especially focused in our recent studies on the iron K alpha lines, so we can determine the oxidation state of the iron in our system. What do we need to do first? So first, I showed the picture of the belt speed of the tape drive, and first we need to check whether our crystals are still suitable for anaerobic crystallization. And therefore, we filled our reaction chamber just with helium and collected an anaerobic data set. Then we also obviously want to see whether we have turnover in crystals. And we were able to collect a product structure to high resolution after exposing the crystals, just taking them out of the glove box, having single turnover, and the product is indeed trapped in the active site. And this is a really important feature for this model, because we are using IPNS as kind of a model system for this, to study these 2OG oxygenases. Because we can kind of trap IPN, so the product in the crystal, due to crystal lattice packing, and this enables just to focus the whole reaction on the single turnover step from our crystallized IPNS iron ACV complex and then to our product complex. So we do not need to deal with substrate moving into a substrate, coming into the crystal, and product release. And what we did next, so all experiments I'm showing now were collected from the same crystal slurry, so we can actually compare them. And they were done, basically, we exposed our crystals to oxygen. And yeah, as mentioned just a moment ago, these crystals were all in a big reservoir, anaerobic reservoir. And then we just, after completeness for the anaerobic data set, we just switched on the oxygen chamber, filled the oxygen chamber, and changed the belt speed and collected data sets for 400, 500, and 800 milliseconds. And all of these maps, first of all, look fairly identical. And we were quite surprised about this because we were not expecting that the diffusion of the oxygen into the crystals is so slow. But if we look now at the B factor analysis, we see increasing B factors starting from 500 milliseconds. And it's quite surprising that the B factors seem to rise from on the side of the ACV, where basically on the valine side of the tripeptide. And this is basically where the vacant iron site is. If we come to the 1.6 second data set, the B factors increased quite a lot already. And after having a look on the 1.6 second data set, we saw the actual first real difference in electron density maps. I calculated some isomorphous difference maps shown here. And in the vacant iron site, you can clearly see some green density. And the best model reflecting the scattering is actually an iron 3 bound superoxide or an iron 2 oxygen. And unfortunately, the iron is not bound, the oxygen is not bound completely. So we still have in the 1.6 second data set 20% of the non-reacted conformation of ACV. If we are going to later time points now, still all these crystals were collected just by changing the bell speed from the same reservoir. We see that the map's getting more and more blurry. We have disorder in the aminobutyric acid side chain. We have disorder for the valine isopropyl. And I tried to model in just this conformation, ACV conformation C, but this model was not reflecting the scattering properly. So I decided also to model in second conformation where I have the cysteine side chain slightly flipped. And this basically shows the superimposition of both. And we kind of have first a disorder of the complete valine isopropyl group, but then also a disorder of the carboxylate. And we can also see that there's quite a lot of flexibility now in this carboxylate region, which is important for the next slides. As mentioned before, we were also collecting simultaneously to the data collection emission spectroscopy. And here are the iron K-alpha lines. And we see of the 1.6 second and the anaerobic data set, and there's small differences in magnitude and shape of the signal. And if we look at the difference spectrum between both of them, this is a blue one. We see that there's a slight positive peak in this region here. And if we compare it to the standard solutions, so the iron 3, iron 2 difference maps, we see that this is only occurring, and this most likely this highlights an oxidation of the iron 2 to an iron 3, highlighting the iron 3 superoxide formation. We did this obviously for all the other data sets too. In the data sets where we don't see any positive density coming up in the free vacant iron side, they're basically identical to the anaerobic data set. In the data sets where we see positive density and where the oxygen or superoxo was modeled, we see positive peaks. And the 3-second data set is identical to the 1.6 second data set. So we kind of have an oxidation from iron 2 to iron 3 due to oxygen binding. To summarize this again, so in the ground state structures, we have ACV, oxygen binds. Due to the steric clash, the valine group gets pushed away a bit, and the isopropyl groups were omitted in the models because we couldn't model them. In the 3-second data set, even the complete isopropyl group was not visible anymore. And in the second model for the 3-second data set, even suggests a flip of the cysteine side chain to bring these CH bond closer to the oxygen for stereospecific cleavage of the bond. And yeah, simultaneously we collected these emission spectroscopic data. We also had a look on the global B factors of these data sets. and we saw surprisingly changes in two regions. So on the one hand, in the heat map of the anaerobic data set, there's pretty much nothing going on. And then I'm just highlighting the 800, 1.6 and the 3-second data set here. And you can see that these particular regions have increased. The B factors have increased drastically. And whereas the C-terminal region stays high in the product complex and goes, the alpha-3 region goes back again to the B factors. So the alpha-3 region gets more ordered. And we were wondering what happened here. And I had a careful look on the 1.6 second and the 3-second data set and calculated difference maps. And there was a lot difference density in these regions, in the alpha-3 region visible, but I was not able to model these differences in. So what I did next, we collected several structures of single crystals, so single IPNS crystals exposed to oxygen, and freeze-trapped them, did single crystal synchrotron reaction. And in most of the crystals where we see clear positive density next in the vacant iron site, there was also increased B factors. In all of them, there were increased B factors of the alpha-3 region. And in some of them, it was obviously actually possible to trap a second conformation. So due to oxygen binding in this region, this valine isopropyl group gets pushed away. The methyl groups are not modeled in this, are not modeled because they're just not visible. And also the carboxylate gets slightly disordered and triggering interaction with the serine 281, triggering the serine 281 pushing away by 1.2 Armstrong. So in these O2 complex, there is a second conformation forming of this helix, of this beta 11 region here. And the ejectant residues, here the glutamine 280 and the valine 282, are interacting with the alpha-3 region, causing a second conformation in the cryo data sets. There are quite a few interactions involved in this. I just want to point out that on the top of the helix, so it's probably somewhere here now, there is this interaction between histidine 62 and tyrosine 90. And we see that they're interacting, and the tyrosine 90 is ejectant to tyrosine 91, which indeed also interacts with the aminotypic acid amino group. And we believe strongly, because this amino acid side chain goes up in the other data sets, the B factors go up, it's getting disordered, and it's ordered in the product, that this might cause after turnover, this is getting it more ordered again, and this might cause tyrosine 91, causing tyrosine 90 to push helix alpha-3 back to its ground state structures. We are still working on it to prove this, but I want to just, because it's quite a lot of mechanistic details, I just want to show a short presentation. So we are starting with the anaerobic data set. We have O2 binding, we have the second conformation forming, triggering this beta 11 part moving back. This is happening. We are pushing the serine 281 by 1.2 Armstrong back. And then there are a lot of key residues which are triggering alpha-3 also to rearrange. And as mentioned before, we have this interaction here with histidine 62, tyrosine 90, and this is kind of involved in, after turnover, we have after turnover, we have this order again, and the tyrosine 91 is pushing the helix back to its ground state structure. So we see the ground state structure in the product complex. You have also seen the active site volume extension, which happens transiently. I want to highlight this here shortly. So we have our substrate structure where this region of beta 11 is in conformation 1. We have the product structure where this is again in conformation 1, and we have the intermediate structure where we have a transiently increase of the active site volume. Then we had a look whether this is also happening with nitric oxide. So we used nitric oxide as oxygen surrogate, and indeed we see the same rearrangement and B factor changes after nitric oxide binding. And although they are slightly different orientations of nitric oxide and oxygen, but as you can see here, they both flip a bit to the side, and the substrate conformation is taking the same conformation. We use these nitric oxide now to see whether these results are relevant in solution. So what we did is introduce the mutation in the, instead of serine 55, we had a cysteine mutant here. We labeled it with 19F by using bromo trifluoroacetone. Then we wanted to use this one for mechanistic studies in solution by NMR. We titrated in, first of all, iron. We see a NMR shift of the 19F signal. Then we add the substrate. We see again a shift in the substrate, in the 19F signal. After adding nitric oxide, we see again a small shift, which kind of supports the data we see in solution. And also we are adding nitric oxide to the iron complex, and there we don't see any shift compared to the iron. And so we definitely need ACV to trigger these shifts to happen. And and obviously also the gas which is binding to the active site. And then at the end, we also wanted to see whether it's working also with other metals. Cadmium is not known to bind nitric oxide, so it's our negative control. We used cadmium, titrated in cadmium and ACV. We see a change in the signal. If we add nitric oxide, we don't see any change, and the same for the cadmium nitric oxide complex. I also repeated this a few times with some crystals which I got with cadmium in the active site, and in all of them, it was not possible to add nitric oxide to this active site. Why is this relevant? So we believe that the correlated motions observed with IPNS are also likely of general interest for the other 2OG oxygenase family, because they all have a similar fold. And we hope kind of that future work will show this, for example, for Rabe, which is involved in DNA repair, or a PHT2, which is responsible for the for human hypoxic response. To summarize everything, so we saw that local and global motions induced by O2 binding enables IPNS to catalyze this beta-lactam formation. We are working on trying to trap these key intermediates. We have achieved now the substrate complex at room temperature. We have achieved the product complex, and we also achieved the O2 complexes. And we are still working on the iron 4 oxygen intermediates. And we hope to get it done soon, but due to COVID and traveling restrictions, it's kind of not easy at the moment. And we are hoping to see that alpha 3 and beta 11, which are present in multiple other 2OG oxygenases, that we see similar findings in the other 2OG oxygenases. And we hope that this work helps to understand how these macromolecular catalysts are working. At the end, obviously, I want to thank several people from the University of Oxford, Chris Schofield, who's my supervisor, then all the people listed here who are always doing a great job. Then from Diamond Light Source, Ellen Orwell, Pierre Agata, and Robin, and from the Lawrence Berkeley National Laboratory, the people who are providing the X-ray emission spectroscopy in the tape drive, Jan Kern and Jonko and Witte, which are the PIs, and then all the people involved. And I want to also thank several funding sources, BBSSC, Wellcome Trust, the UK XFEL Hub, and yeah, the Leopoldina for funding of my postgraduate stay here in Oxford. Yeah, if you have any questions, I'm happy to answer.
Super, thanks.